Anyway.
All right, I think we have a quorum. Thank you, everybody, for coming to our new RIPS, the Genome Editing and Technology Application Series. This is a new series that's born from a desire to reinvigorate our RIPS, B, the strategic planning we had, and specifically the recommendations of the Genome Editing and Stem Cells Committee to have more RIPS focused on these things. And then C, conversations around building a Center for Genome Surgery that would be a partnership between the IRM and the Innovative Genomics Institute. So all of those lines of thought are coming together, and I'm especially grateful to Cassie for putting this amazing series together, along with our faculty consultants, Kyle, Julie, and Holger. And please let us know if you have feedback, if you have suggestions for additional speakers. We're really trying to invigorate a community of people who really care about genome editing and stem cells, potentially for basic science applications and potentially for therapeutic applications. So, and what better person to present our first session than Kyle Cromer? Take it away.
Okay, thanks. Okay, so kicking this off, as Tippy mentioned, IRM strategic vision, which I was part of the committee helping to try to generate this, is discover, engineer, heal. I'm kind of on the engineering side. A word that we say a lot in our lab is, treating these cells as if there's things to be engineered. Where we're starting from is, and I'm going to focus this talk on genome editing, which I believe is, one of the most powerful tools we have in a greater cell engineering toolbox. And if we think about these CRISPR therapies that have come out, the very first thing that people wanted to do with CRISPR therapies is take patient cells and make them wild type again, which has been highly effective, especially for low-hanging fruits like sickle cell disease. But as we've acquired these tools, we now realize that there are additional bottlenecks. They're preventing widespread adoption. There's the extremely high cost, the toxic chemotherapy required to clear space in the bone marrow for these cells. And I think that really just highlights an issue with the cells is that original paradigm of taking patient cells, isolating them in these time, cost-intensive workflows, making them wild type again in the best of circumstances and transplanting them back in. Best case scenario, you have wild type cells. And so that's helpful. That's better than having diseased cells, but they're not that therapeutically potent, not any more than nature made them. And so we think a lot of, in our lab, what are your cells not doing that you want them to do more of? And so, well, wild type cells, they differentiate normally. They don't really home to the bone marrow any better than wild type cells, don't vascularize perhaps when you transplant them. They can be immunogenic, they can lead to adverse events, and we have little recourse if these things happen. And so in my own lab, we really think about these cells as tunable, engineerable objects. And we believe that we have, just as evidenced by the CAR T cell space, you have T cells in the body that are not doing the job that you would like them to do. But now we have the tools and knowledge to engineer them to do that thing that they're not doing. And so that can be to take control of cell fate decisions. In the context of disease, oftentimes you just simply have one broken differentiated cell type. And so how do we engineer our stem cells to make more of those? How do we engineer our cells to better home to the bone marrow, perhaps? How do we engineer them to better vascularize, engineer to be off the shelf in these ways that would be immunogenic and could actually help the scalability and accessibility of these therapies? And then also just some recourse if there's an adverse event. For instance, putting in small molecule inducible safety switches. You want an emergency brake in your car, even though you don't want to have to pull it. And so before I talk about genome engineering, I have to talk about the foundation for this, the genome. There was a 50-year gap between the discovery of the double helix structure of DNA and when the first normal human genome, consensus human genome, was established in 2001. Coming on the heels of this was, for the next 10 to 15 years, really felt like a golden age of driver mutation discovery, of which I played a small role. But really, more importantly and more broadly, this allowed us to think about reading the genome as a Microsoft Word document. Well, now with CRISPR and coming out of grad school, when I was doing a lot of sequencing, I became frustrated because all of this information was great and it was a great starting point, but a starting point for something else to follow it. And I wanted to do something that was actionable with all this knowledge that we now had. So I wanted to think about editing the genome as a Microsoft Word document, and the timing kind of synced up pretty nicely. As I was ending grad school, the CRISPR technology was first emerging. And so it was obvious this is what I wanted to do afterwards. as we think about genome editing, I first have to mention what happens normally in these cells, because all of the genome editing tools are essentially hijacking the cell's endogenous DNA repair processes. And so double-stranded breaks are happening to all of us right now for a variety of reasons. Cells invest very heavily in preventing and repairing double-stranded breaks. They will detect and activate this cell cycle checkpoint, the cell cycle pause until these double-stranded breaks are repaired. And if they remain unrepaired or misrepaired badly enough, apoptosis or senescence happens. And so really when we think about a double-stranded break, I'll generalize, and we talk about non-homologous end joining as being this error-prone repair, just essentially slapping these two double-stranded breaks back together. But when you have a double-stranded break, the ends of these DNA strands are quite brittle, and so you can have gain or loss of different base pairs. And this is really helpful for disrupting certain regions of the genome. We also can have perfect repair whenever these double-stranded breaks naturally occur. And this is using the sister chromosome as a repair template to allow, even if you have some of these insertions and deletions happening with the brittle ends, the cell's endogenous repair processes, homologous recombination, or what I'll refer to as homology-directed repair in the context of genome engineering, can yield perfect repair of this region. And so Cas9 essentially leverages these endogenous DNA repair processes to specifically scan the genome, essentially to extend the analogy of Microsoft Word document. It performs the control F function. It's a protein that can latch on to a user-defined guide RNA, scan the genome until it finds a perfect or near perfect match, and then it'll initiate a double-stranded break where I've highlighted here this red line. It operates off this protospacer sequence. So this is the user-defined sequence that you feed in. And typically a PAM sequence, protospacer adjacent motif is what people will call. And so when somebody says CRISPR, they just say CRISPR, they probably mean CRISPR version 1, and this is SP-Cas9. And so the PAM in this case for these Gen 1 CRISPRs are an NGG. And then we define the sequence of the guide RNA, the protospacer is 20 base pairs upstream of that, and the double-stranded break happens 3 base pairs upstream of the NGG PAM. What then happens if you initiate these double-stranded breaks is you can have either error-prone repair, which results in disruption of the site. And so this is how it was used first. It's actually how the first FDA-approved therapy operates. It breaks the element that silences fetal hemoglobin, so you can reactivate that double negative of breaking a silencer, reactivates fetal hemoglobin to compensate for the broken beta globin in the context of sickle cell disease with these optimized editing protocols. I mean, these are some tools that when I first got my hands on them, I realized they are not overhyped. They are easy to use and they work well. You can end up yielding desired genome edits at greater than 90% frequencies with optimized protocols. This is, again, all ex vivo editing for the most part until we start talking about in vivo delivery at the end of the talk. And so with this, the first FDA approval was achieved for CRISPR-derived therapy for sickle cell disease in December 2023. However, a caveat, if we're thinking about just using Cas9 version, CRISPR version 1 to break things, most diseases are caused by broken genes. And so there are just few examples where breaking more things can be curative. Sickle cell disease is one of the very rare examples where that can be true. To do something that would be corrective, you need to fix the mutation. Homology-directed repair is much more flexible, but also you have to deliver more machinery. So what this means is you do the control F cut function with your Cas9 directed to a region of the genome that you want to insert something there. And then you provide a repair template in a variety of methods, but at least for primary HSCs, we have to do it using an AV repair template. And I also know Justin Equium is a huge fan of these in T cells. It's a great way to edit 1 to 4,000 base pairs, essentially put 1 to 4,000 new pieces, new sequence anywhere in the genome. So essentially, we can use this as a platform, Cas9 AAV, to ask the question, what new code would you write and where would you put it? And this is extremely, extremely precise. We can knock in entire cDNAs in frame into endogenous genes, leverage this to allow these transgenes to be driven by endogenous promoters. However, in the case, this is probably happening most of the time you have a double-stranded break is no editing or perfect repair. And so if perfect repair happens, which is probably happening constantly, then your Cas9 is still finding its perfect match at that site, cutting, perfect repair, cutting, perfect repair, cutting, perfect repair until something stops the reaction. So what can stop the reaction? Well, an indel can stop the reaction. And then that disrupts your guide RNA sequence. And so that now this region is no longer homologous enough to initiate, again, recutting. Also, if you were to perform your ideal homology-directed repair process, that will also stop the reaction. Or if you have perfect repair frequently enough, at some point your Cas9 will degrade. And so this is how we think about this. And there's some strong evidence to say that the most likely outcome of a double-stranded break is perfect repair. But these are the end results that stop the reaction that actually end up becoming enriched by the end of the editing. Also, I would also say, I guess we have a Q&A at the end, but people should feel free to ask questions. Yeah.
Some people include like a base pair mutation in the guide RNA, like they change the sequence of the DNA, non-synonymous.
Yeah.
So to prevent this recutting, can that be a strategy?
For your HDR template is what you mean?
Yeah.
Yes, oftentimes. So the best way to eliminate recutting is certain mismatches will be well tolerated. The further you get back from your PAM, the more tolerated a mismatch will be. However, if you really want to break any additional recutting, the PAM is the thing that is best to disrupt. If you have an NGG PAM editing tool and you disrupt that with your repair template, so you can design it in that clever way, you're not going to have any other cutting. I think of it a lot as a zipper on your jacket. The PAM is the start of it. And then as you zip up, the further you get to a full jacket zippered closed, the more likely that double-stranded break will happen. And so if you think about where mismatches are more tolerated versus less tolerated, the closer to the PAM or right after the PAM is going to cause that zipper to catch more likely, and then it'll just keep scanning. But if you fully zipper, if you want to create an intentionally bad guide RNA, which sometimes we do for basic science purposes, I'll create a mismatch at the very, very tail end, and you're going to get off-target editing activity if you do that. Okay, what does this look like? And, in what format are these editing tools delivered? Well, the very first ways are these ex vivo strategies. I guess first in cell lines and then primary cells, primary T cells, primary HSCs. Ex vivo delivery typically happens via electroporation or liquefaction. Electroporation is even being used in the clinic. And so in that clinical ex vivo editing workflow, CRISPR Therapeutics, Vertex, that was FDA approved, this is electroporation of a Cas9 protein pre-complexed with the guide RNA delivered to the cells that you've isolated from the patient. You then have to give them chemotherapy, retransplant those cells. And so it's a complex process, but oftentimes what we're doing in the lab is trying to be in a research grade way, mimic what's being done in the clinic. However, in the future, where the entire field seems to be pointed in the direction of in vivo delivery, so you skip all of those time and cost intensive steps of pulling cells out, editing them, giving somebody chemotherapy to clear out space in the bone marrow. Instead, we could just, at least from an HSC centric point of view, edit HSCs where they sit in the bone marrow and skip all those steps. And that can be done using lipid nanoparticles. Those lipid nanoparticles can most often carry mRNA coding for your editing tool and a guide RNA. It can be done using VLPs, viral-like particles, EDVs, envelope delivery vehicles, or AAV. A caveat with AAV, even though that was one of the first ways people were doing in vivo delivery of CRISPR tools, is these Cas9 editing tools are quite large. And so to be able to deliver them by AAV usually requires you to split them apart. And that's definitely going to lower your in vivo editing efficiency. Once you've done this, whether it's in vivo delivery, ex vivo delivery, we then do a local PCR of the region that we expect to be edited, and then we can run Sanger or targeted NGS. And just what this looks like from looking at Sanger traces perspective, here's an unedited trace, and then here's what it looks like if it's been edited. It's these insertions and deletions. I can even tell from this Sanger trace. We have a cut at right around the place that we expect it to be, 3 base pairs upstream from the PAM. And again, you can actually see this guide RNA is targeting the minus strand instead of the plus strand. They both work just fine. And I can also tell from the Sanger trace, we're reading it with a forward primer because it's clean until you get to that cut site, and then it's highly disruptive because you have this mixed bag of insertions and deletions, creating this chaotic mess afterwards. So this is really the calling card of, how do you tell the difference between genome editing or just a bad Sanger trace? So now I'll talk about these next generation tools that we started using in the lab. They're called base editors, and they essentially still do the control F function, but they can lead to substitution events without creating double-stranded breaks. So they're either a nickase or a dead Cas9. These new next generation tools, nothing's better at doing the control F, user-defined guide RNA work that a Cas9 is done. And so what happens in these next generation tools, they take that backbone of Cas9 to perform the control F function. They'll then neutralize its ability to do the double-stranded break, either making it a nickase or a completely catalytically dead Cas9. But then they fuse interesting things to it, so it can drag really any user-defined protein to a novel place in the genome. And I'll talk a little bit more about how else this is being used. But we've been really impressed by base editing tools. Again, this is a set of tools that I think the hype is real. The very first time we started using them, they worked well. They were easy to use. That's why you see so many publications on these and why it's a really hot area of research right now. This is layering on additional complexity. So I know in my lab, we often ask, okay, what new code would you like to write and where would you like to put it? But then immediately we start talking about how we operate within the constraints of the editing tools. Cas9 you need a PAM nearby, and then you need to define a sequence that ideally is not occurring, a repetitive sequence somewhere else in the genome. A base editor has additional constraints. You need a nearby PAM, but then you also then have your editing window where you need a base pair that is the desired base pair that can be changed to one specific thing. And there are additional constraints here. The two main classes, so it's tethering this deaminase, it'll do control F, this nickase or dead Cas9, and then it's tethered to these deaminase domains. Adenine base editors convert A to G within this window, and so it would likely convert this A to a G. You also have this risk of a possible bystander A that's within your editing window, or a CB, cytosine base editors convert Cs to Ts. And so if we look at the same editing window, this is likely to be changed at some high frequency. And these, if that's a desired change, and these are potential bystander edits. And while it's fairly predictable, they don't follow those precise rules. Sometimes when you shift your guide RNA around, you would think that it's going to be really obvious. If you have that center of the window right over your desired base, you're going to get most of your editing there. It's not always predictable, and there's no substitute for actually just testing your guide RNAs in combination with your base editing tools. Again, also, this is just an example targeting the plus strand. You can flip this around and target the minus strand just as well. So here's an NGG sequence in the bottom strand, then you can go upstream of that on that minus strand and feed that in as your guide RNA. And then we're looking at maybe that's a C to a T change that could create this G to A change at the plus strand. And there's no difference between targeting either of those. And again, for the base editors, we follow those same rules. If you have no editing or perfect repair, we're going to allow this base editor to keep going in and keep doing these changes, keep making these changes until something stops the reaction. And some of these base edits, that will stop the reaction, because then the guide RNA is not such that perfect match that it will continue to find that region of the genome. And so the great thing about these base editing tools is they avoid double-stranded breaks, but can lead to guide RNA independent off-target effects, which are a little scary. We haven't exactly observed those, and I think once there was base editors version 1, there now are base editor version 9. And so they're constantly being improved through directed evolution by experts in the space like David Liu. Also, there are multiple base editing clinical trials going on right now with extremely promising results. However, I would say most disorders are caused by loss of function variants scattered throughout a gene. And so unless you want to go in and pick off one mutation at a time, which is absolutely feasible as indicated by the baby KJ story, it is a time and cost intensive process. And so there are limited examples where a single base edit could treat the majority of patients. Also, right now, there's a bottleneck regulatory-wise. The FDA currently views the same formulation with different guide RNAs as a different drug formulation, but that likely will change. And just in terms of what do the base edits look like, so this is work led by Kuhn in my lab, a really talented postdoc. When we were first doing this, we were getting okay base editing. This is about 30 to 40% of the desired C to T change with these earlier versions of base editors at 3 different sites. So they're very flexible, but we were just getting the majority of our cells not containing the edit that we want. But then through really laborious optimizations, We couldn't optimize the mRNA itself. You have to highly modify it for it to be well tolerated in primary HSCs. Whereas for a cell line, you could just use a plasmid and that works fine. T cells are probably a little bit more tolerant of, not these perfectly highly modified mRNAs. Also, another thing is, at least when we started the work, and maybe and a little bit more now, most companies were not selling base editors. There were so many different varieties out there. There's like version 9. It's also just not as common as Cas9. And so if you want to do base editing, you had to make them in-house. And so we really started out doing that. Right now, there are companies that are starting to sell these, and I'm sure they work quite well. I'd have to check to see what sort of modifications they have. But then after that, then you have to optimize the delivery. Whether are you going to get high editing frequency really depends on how good is your electroporation. So there's optimization there. And then when we started this work, because these base editors were coming out at a high frequency, there was a novel base editor that was evolved over the course of these optimizations. So linking all these things together took us from 30 to 40% of our desired edit to 95%. And so when you find a good guide RNA and a nice base editor, they can work at extremely high frequencies. For a long time, it used to be that ABEs were the first class citizens and CBEs took a back seat, but now with these optimized CDs, they work just as well as ABEs. And then we've even, because we're getting highly efficient multiplexing and you're not creating, or highly efficient editing at single sites, and you're not getting double-stranded breaks, we now started multiplexing these. You can do 2, 3 edits simultaneously in primary HSCs, and this is a really exciting emerging story from my lab. Also, just thinking about tethering other interesting things to your Control F Cas9 backbone, you can have CRISPR activators. They do Control F and transient activation, Control F transient repression. If you put inhibitory domains there, and these also work really well. Ben in my lab has really pioneered these and optimized them for primary HSCs. So primary HSCs do not express NGFR, but they do after you add a CRISPR activator. They also normally express CD45, but you can temporarily knock these down. The caveat is they rely on constant expression of these tools, which is great from a basic science perspective, but from a therapeutic standpoint, constant expression of a ultimately bacterial-derived enzyme is likely going to cause immunogenic problems. What's more promising that we don't have experience with, but certainly seems like the right direction for these tools is a CRISPR off strategy. And so this is like CRISPR-I, but it can do hit and run modification and then lead to Control F and long-lasting repression. And that was some technology that was, to my knowledge, first pioneered by James Nunes at Berkeley. And then there are just emerging systems. People pay a lot of lip service to prime editors, but to be honest, they're extremely difficult to use and they're not very efficient. And when clinical efficacy hinges on maximizing your editing frequency, I just think that prime editors are overhyped. However, this is the worst that they will be. They're only going to get better, or there will be something that does what they do, but just better, but is fundamentally different. And so these future systems, the goal of all of these future systems that I'm showing right here, prime editors, these recombinase-based systems, is how do you make small and large changes, even large integrations, without double-stranded breaks? And so that's what everybody is trying to move towards right now. David Liu's working in that direction, Ben Kleinstaver and others. There are a lot of really great people working in this direction, and I think that it will be a solved problem. But right now, the existing tools are tough to use, and you don't get high editing efficiencies with our experience. Now just thinking about, okay, great, we have these tools. How do you use them? And so, well, the original paradigm of taking patient cells and making them wild type again, you can use the tools for disease correction. One of those major strengths is that 20 nucleotide guide RNA provides highly specific targeting. If we just were a monkey at a typewriter and write 20 different nucleotides in a string, chances are that 20 nucleotide sequence is not going to occur even one time in the human genome. And so 20 nucleotides is specific enough that you can target just one region of the genome as long as that region is not repetitive. We also can distinguish one single guide or one single SNP difference in your guide RNA is enough to take your editing strategy. Here's unedited cells. Here are edited cells. This is, again, a Sanger trace read in the reverse direction. So it's clean up until the cut site and then messy after that with all this mixed bag of insertions and deletions. And we were confused as to why one of our donors gave us no editing. But then when we did our Sanger, we realized that a homozygous mutation, even towards the back end of your guide RNA sequence, and it completely obliterated our editing frequency at that site, really just highlighting how specific just even a single mismatch is enough oftentimes to completely eliminate editing frequencies at these sites. And so then you can leverage this. I know Bruce Conklin is doing some really cool stuff with allele-specific editing. Because they're so specific, you can design a guide RNA that's distinguished between patient populations and only create a disruption in the mutant or, and perhaps a mutation-linked allele. So in this case, for polycythemia vera, like 97% of folks with polycythemia vera had the exact same dominant JAK2 point mutation. It's gain of function, it's heterozygous. However, you can't just go in and knock out JAK2. JAK2 is really important, so Cas9 disruption is bad for the cells. What we did is, okay, because we can distinguish by just this one single base pair in our guide RNA sequence, we designed a guide RNA that was targeted towards the mutant allele. Ideally, it would cut the mutant allele to deactivate it and not disrupt the wild type sequence. And that's exactly what we found in these iPSCs in collaboration with Matt Porteous, led by a really talented med student named Freya. We see when we have our wild type guide RNA, we get really nice editing in our wild type sequence, no editing in the mutant line, and vice versa. This editing was not as high as we would have liked. And so actually then we paired that with a downstream edit that was common in both, and we were getting high frequencies of deletions. That's one thing that Bruce Conklin is leveraging, is it's counterintuitive. If you can take two kind of lowly active guide RNAs, even like 30% at a single site, and that was kind of what we had because we're so constrained with where we put this guide RNA here in the context of this mutation. If you pair two kind of low to medium activity guide RNAs, you can actually get an extremely high frequency of deletion between those two sites. And I think that's the anecdotal evidence that's suggesting that you get a lot of this perfect repair until something stops the reaction. Well, if you're getting kind of that low level, constant cutting, perfect repair, recutting at two different sites that are nearby, one thing that can stop the reaction is deletion and loss of that region. And so we see that actually become, you can introduce very precise, exactly as you would predict, deletions using two guide RNA strategies. Also, we leveraged this in other work, this single SNP different distinguishing guide RNA sequences. We wanted to knock a beta globin into the alpha globin locus, but we needed to distinguish using the precision of Cas9 between our two alpha globin loci, which are extremely homologous. The entire coding sequence and all of intron 1 and all of the 5' UTR are exactly homologous. The reason we need to distinguish them is because we didn't want to cure beta thalassemia, but cause alpha thal in the process. And so we tested a variety of guide RNAs that were exploiting just these tiny single SNP differences between these two. And we found two guide RNAs, one that was extremely specific for HBA2 and didn't cut an HBA1, and vice versa, one that was extremely specific to HBA1 and didn't cut an HBA2. And then we leveraged this to specifically integrate HBB into the HBA1 site while preserving your ability to express normal adult hemoglobin as a one-size-fits-all strategy for beta thal. Also, we're using the tools for not just disease correction, but also for basic science. These have an extreme power to shed light on natural biological processes. The value of CRISPR screening in the T cell field cannot be understated, and I think there's a great opportunity to start applying these tools to primary HSCs. The great strength here is the modularity of these guide RNAs. You can just swap out a new guide RNA and target a completely different locus, and they're also really amenable to high throughput screening. The way that this looks and the way that we're using it in my own lab is take your primary human HSCs, so trying to most closely mimic natural biology, that is more difficult to model in cell lines. Deliver your guide RNA pool, however small or large this pool might be. And then what you can do is, in this case, deliver your editing tool by electroporation after delivery of your lentivirus. And that can be a base editor mRNA, it can be Cas9 or RNP. They can then go and grab that guide RNA that's being expressed from your lentiviral construct. You can even then put a puromycin resistance cassette in your lentivirus and just make sure that you're only working with cells that got guide RNA. And then we can do, in this case, we did a differentiation. And this was a base editor screen that was run by Ben in my lab. This was over 1,100 different guide RNAs scattered tiled across the EPO receptor locus. We get really good representational library. And from that base representation, we can say, okay, over the course of this differentiation, which guide RNAs were enriched and lost. And so what we see is across replicates, these are highly reproducible, this like log2 fold change enrichment and depletion, indicating that the changes are consistent. Unsurprisingly, there are many, many, many more ways to break a gene than to hyperactivate it. We were looking for the hyperactivated ones. Again, thinking about the base editing tools and their constraints, we think that base editor screens they're just inherently limited because you need a PAM that's nearby. You then need an ideal base pair, either an A or C in your editing window. You probably want to avoid some of the bystander editing, or unless that's maybe a feature. And then some guide RNAs are just low activity. And so there are a lot of guide RNAs that probably just drop out for no reason, and a lot of missense mutations that you just can't make if you're only making A to G changes or C to T changes in the plus or minus strands. So while helpful, this was a great proof of concept that we can actually do these. They're highly reproducible, identified gain of function, loss of function edits. But what we wanted to do after that is, I don't think there's anything quite as elegant as just a genome-wide Cas9 screen. And so Ben just completed the first, in my lab, genome-wide Cas9 knockout screen in primary HSCs using a 70,000 guide RNA library. In order to do this, you're a little bit constrained, and for the long time, we were constrained of, you need a lot of cells at the start to be able to deploy a 70,000 guide RNA library with sufficient coverage, enough cells that each independently have the same guide RNA that's inserted. The number that we started out with was 35 million, but then we differentiated these into red blood cells and sampled them throughout. And Ben also was able to isolate the pyrenocytes, that the nucleus that's been spit out of your red blood cells, to see what guide RNAs are enriched or depleted in those populations, indicating activators or repressors of the process. And so we're really excited to get those results soon. We can also use these tools just for engineering. If we think about, okay, well, we can better understand, maybe identify engineering strategies to enhance native processes and correct patient mutations, there's also just this major strength that we can treat DNA like computer code and go beyond this correction of disease-causing mutations, engineer new properties into cells. And so one thing we've become really interested in my lab is taking control of cell fate decisions using genome editing, especially in the context of sickle cell and thalassemia. These patients are otherwise healthy. They just have broken red blood cells, to oversimplify. And we spend all this time, effort, and heartache maximizing our editing frequency in HSCs, giving patients chemotherapy, so we maximize engraftment of those edited HSCs in the bone marrow. And then your sickle-corrected HSCs become sickle-corrected T cells, B cells, and macrophages, as well as red blood cells. So we wanted to write some new code in our HSC that didn't alter its long-term stemness, but did coax it to produce more red blood cells. And so this is a story that many of you have probably seen, but we took inspiration from nature, this Finnish Olympic cross-country skier from the 60s. We then used the precision of genome editing tools, and we installed it in a variety of ways. We did it with Cas9, we did it with Cas9 AAV, we did it more recently with base editors, and they're all highly effective. This is a really potent mutation, and it really leads to an extreme hyperproliferation of your cells that have this edit. What we can also do is just go a step further. As we started modeling this EPO receptor natural variant, what we also said, we started to gain insight into how the EPO receptor worked and became interested in, well, could you perhaps engineer a small molecule inducible EPO receptor? And so this work also in collaboration with Matt Porteous, and suffice it to say, yes, we can. I don't need to go super deep into this story exactly, but what you can do with these genome editing tools is you can write brand new, not found in nature sequences using these synthetic elements. You can then use the precision of genome editing to install them at different places in the genome. And we found throughout this process that installing these non-native sequences at different places in the genome, allowing them to be driven by endogenous promoters or exogenous promoters that we provided, even though it was the same ATG to stop codon sequence that we're installing, they can behave quite differently depending on the expression profile that your synthetic element assumes. But because this Cas9 AAV editing is so precise, you can explore those sort of questions as well. And then I think what probably many of us are also interested in, and I know that it was identified as a need area for the IRM, is delivery. Dave Schaefer likes to say in the field of gene therapy, there are three problems, and it's delivery, delivery, delivery. So I'll steal his comment there, but really for genome editing, that's what we're faced with as well. While disease treated by bone marrow transplant can be cured by ex vivo genome editing, and it's long been viewed as a low-hanging fruit, many diseases don't have stem cell populations you could just pull out, edit, and put back in. Huntington's, Duchenne, cystic fibrosis, SMA are great examples of these, and many, many more. And even for diseases that you can fix in this way, this ex vivo time and cost-intensive process, it's really involved, and it inflates the cost. It leads to extended time in the hospitals. We then also, because you're having to pull these cells out, and then they're competing against other wild types or other cells resident in the bone marrow, you need to give chemotherapy. That's a huge safety bottleneck to these unengineered, now patient converted to wild type cells that you retransplant. So Francis Collins even said, when he was back heading the NIH, was on a cure for sickle cell disease, genetic cure would have to be given directly to patients, rather than by treating cells removed from patients and genetically transformed in the lab. That could avoid the resources needed and complications that can occur with ex vivo therapies. It's my firm belief that the only way that we cure sickle cell disease worldwide, it has to be not patient-specific like this, these bespoke therapies. Ideally, it needs to be in vivo genome editing. And so what that can look like is just a systemic injection of very intelligently designed and optimized tools that's a delivery vehicle that, in the ideal circumstance, goes and finds the cell type that you care about and makes the change in those cells directly, where they sit in the bone marrow. And that's the very HSC specific way of looking at things. But whenever we're doing genome editing, your target tissue could be in cystic fibrosis, it could be lung. In these urea cycle disorders, it could be liver. In vivo editing is tough, and it opens up new safety concerns. Really, when you do in vivo genome editing, there's only one desired outcome. You want the edit that you want, no off-target mutations, ideally, and you want that to only occur in the disease-relevant tissue. Why do we need to correct the sickle mutation in the liver? That just carries some unintended risk of genotoxicity, especially if we're creating double-stranded breaks. And then you're expressing these bacterial-derived enzymes in vivo in a tissue that's not going to make a clinical impact on disease correction. And so there's a lot of effort and a lot of great people working on this targeted delivery of delivery vehicles, payloads that are only expressed in the cell type that you want editing to occur in. And so there are major strides being made in this area. And I think that I'm really optimistic about the future of in vivo genome editing. To show some data to back it up, I don't see Tippy's lab on this schedule, so I took the opportunity to give credit where credit's due, but we need to talk about delivery. And so in vivo delivery is possible with, well, kind of originally AAVs. There are now VLPs and EDVs that have been developed. I'll just say that I'm most optimistic about LNPs. They can be manufactured at scale. They're non-immunogenic, so you can even redose them. And the editing efficiencies are good so far. And so the McKinsey lab has expertise and some great folks working on these reporter mouse models with rapid fluorescent readout of whether you achieve genome editing or not. And so there's this traffic light reporter mouse that was developed at University of Washington that has this, it's a GFP right here, driven by an CAG promoter in the transgenic cassette. And there's an RFP downstream that's out of frame. Well, because Cas9, it's cleaner to turn on a fluorophore than to take something that's like GFP on and target it that way and look for your GFP negative population. It's cleaner to look for a double-stranded break that turns on a fluorophore. And so this was the clever way that they created this traffic light reporter mouse, where you have a downstream fluorophore that's out of frame. You then intelligently screen and choose guide RNAs that happen to restore that downstream reading frame. And so then a double-stranded break turns on a downstream RFP. And so in some recent work, this is in utero injection, but they've been doing postnatal injections as well of these LNPs from an industry collaborator. It's expressing it then delivers Cas9 mRNA plus a guide RNA targeted to this site upstream of the RFP and getting clinically relevant genome editing frequencies in vivo in these mice. The number that's always put out there for sickle cell disease and thalassemia is 20 to 25%. And so this is really impactful potentially. Then, because base editors are such an amazing emerging tool, they also have a base editor reporter mouse that was first developed by Anishka Chekowitz and David Liu And this is a great way to take something that's GFP off. There's a premature stop codon, but you can go in with an ABE and change that premature stop codon to allow GFP to be read out. And so that gives you really good editing. Again, there's no place easier to edit in vivo than the liver. HSCs are more challenging, which is why this data was so impressive. And then more recently, we can use the traffic light reporter mouse as a readout for in vivo HDR as well. And so here's the negative control. Here's just when you deliver a Cas9 LNP, and so we get really high frequencies. This is in the liver of this double-stranded break that reactivates the downstream RFP. And then when we provide an AAV repair template that actually fixes what's upstream of the RFP is a heavily broken GFP, we can restore the function of that. And so this was really, really encouraging that we can do knock-ins, precise knock-ins in vivo. And this could really open up the door, for instance, for a urea cycle disorder where these really severe early onset disorders, most patients are only going to present with a unique mutation, but HDR would allow you to knock in a cDNA to a safe harbor site to, again, create a one-size-fits-all therapy for folks. Also, highlight of the work that's being done in Tippy's lab is this injection at early developmental time points increases editing rates. There are many reasons to do this. Actively dividing accessible stem cell populations, a low dose goes further. So you could do it in utero, but also we've been getting amazing results just neonatally. However, you have to diagnose neonatally or prenatally to have these therapies ready by the time the patient's born. And the long-term goal is to be able to achieve these highly efficient, highly cell type tropic editing events in adults with disorders. So I think with that, I'll end the talk. There are a lot of people to thank. I showed a ton of different data here. There's Karen and Ben in my lab. There's Beltran, Natasha, and all of the McKenzie lab team. Some of the data that was done most during my postdoc and early on in my time here by Matt Porteous's lab and the great people there. And have to appreciate my funding sources. Grateful to be part of the IRM. And yeah, I think now we can do the question and answer session.
Kyle, there might be questions in the chat too. I think there's a bunch of people on Zoom. Courtney, will you tell us if there's...
Yeah. There's nothing currently on chat.
I have one question about the scenario that you showed where you have two sequences that are very homologous and you're trying to identify guide RNAs that are specific for either one. Once you found some and tested them and do check by Sanger sequencing that you have a certain level of editing efficiency, what's your preferred method of checking any off-target cutting frequencies?
I had that as part of this talk, and it's like a talk unto itself. Happy to give a quick glance. There's so many different strategies for finding your off-target mutations. There are a bunch of Nature Methods papers, CiteSeq, CircleSeq, GuideSeq, DiscoverSeq, all of these. And they're all claiming that you get thousands of off-targets that are only discoverable by that certain method. They're also just time and cost intensive methods. We had a great interest in trying to understand how do you actually find the needles in a haystack, because these claims that you have thousands of off-targets that you're not finding by any other means just didn't mesh with our experience when we're doing this transient genome editing in primary HSCs that have good DNA repair processes. Some of the seq methods are like operating on cell-free DNA with no opportunity to repair itself, stable expression of wild-type Cas9 and cell lines. All of that is, suffice it to say, we found that we try to benchmark all of these different processes and say, are we actually missing them or are they rare? We found they're extremely rare when you do this transient hit and run electroporation of something that affects that permanent change of a Cas9 or a base editor. And they're always guide dependent. It's always homology dependent, at least for Cas9. And so the best thing you can do to make it a manageable problem to solve is there's one tool in particular that we found to outperform the others. It's called COSMID. That is an in silico, just purely homology based on the consensus genome. So there is a little nuance there as well. And there might be things that you could miss based on, patient specific or population specific SNPs. But what you can do there is you can use that. We defined a cutoff score because it'll rank order based on the degree of homology, where your mismatches are. which ones to focus on, and that can usually get you to half dozen or a dozen sites that we then do targeted NGS of those sites, and that's enough to really answer the question for the most part.
Okay, so you do, in short, you have your guide RNA sequence that you input into the CosMed pipeline, spits out your ranked putative off-target sites.
Yeah, exactly. Yeah.
Thank you.
Robert?
Is there a limit to what you could do in vivo? Imagine you wanted to make 6 knock-ins, 5 knockouts, and I don't know, engineer a cell. With that unlimited in many ways, is that going to happen in vivo? And isn't that kind of where we're going?
Yeah.
So I'm just trying to match where people can engineer cells to crazy levels. But simultaneously want to somehow do it in vivo.
I think what we'll see first. So I love the consideration. Like at some point, the tools get so good that it's just our creativity and our imagination that's the limit. But that's kind of a hand wavy statement. And I think we're pointing in that direction. People are starting to do multiplexed genome editing, so simultaneous introduction of different edits. However, what we're trying to do right now is that simplest thing of getting high enough efficiency of a single edit in the desired cell type that we're not quite at that level yet. Like I think we need an FDA approved in vivo genome editing strategy to start, and then we'll see more complicated things. I think overall what we'll see is, and what feels like lower hanging on the tree, is knock-in of really sophisticated synthetic elements, but just a single knock-in site. I think that that's going to be more straightforward in vivo HDR. You could even imagine a library of things that insert into various cell types, and then you pull out the cells that are behaving in this enhanced way or something. I think my gut says that that's going to be easier to do than multiplexed in vivo editing for the foreseeable future.
You just talk about like CAR Ts, right? I mean, they are multiplex manipulating.
Yeah.
Is that happening in vivo? And how far is that happening?
As far as I know, it's always ex vivo. Never are cells easier to do these complicated multiplex edits than ex vivo. And so it's happening right now in the CAR T cell space. It's also happening in the primary HSC space for sickle cell disease. You can correct the mutation, then you can layer on one more edit. But I think multiplex editing for the foreseeable future is only possible when you do this ex vivo editing. We're just not very good at installing a single edit in vivo yet, especially in HSCs that are more challenging. So we'll need that before we can start investigating feasibility of multiplex editing. And then it will matter if you do multiplex in vivo editing, all the editing rates I just showed that Tibia's lab is achieving, they're good, but when you have that kind of low to medium level of editing, if you're doing 2 edits, you're probably just going to have cell populations that have one of the other, and a limited set that have both together. Unless from a technological standpoint, you could more effectively, cleverly pair those. So, and you could imagine maybe an AUV that delivers only your guide RNA. So at least if your Cas9 encounters a guide RNA, it's going to encounter both of them, perhaps.
Question about say you base edit with LNPs. Is there any tropism for cell? Like, is it easier to edit certain cell types than others? Like, say I want to edit L05 in the intestinal stem cells. Is it just like you just plug and play, or is every system every tissue or target needs a new approach?
Yeah. It's been our experience, we haven't tested it super rigorously, but a good guide RNA tends to be a good guide RNA across different tissues. Certain tissues are easier to edit, mainly because it's my belief that you could find a good guide RNA targeting most regions of any tissue that you want. Then you're going to hit your head up against the limits of how good is your electroporation or whatever method you're delivering those tools to that tissue. And that's usually where we take major hits in terms of our editing frequency. If we're trying to edit, like we're really good at editing primary HSCs, people have really refined your primary T cell editing. To move to a new cell type, it's usually inefficient electroporation that needs to be modified. Maybe you need to create some sort of more nuanced, like RNA sensing pathway shielded mRNA in those cell types. But those seem to be solvable problems. And then ideally you would have flexibility with where you put the guide RNA, but that's much less so with a base editing approach where you really probably have the one change you need to make. There's only so much you can shift your editing window. And so it's kind of a matter of, did you find a good guide? Is that guide RNA interacting well with the base editor at that specific DNA sequence? And sometimes it just doesn't, or you can't clean up bystander editing, and maybe that bystander edit is creating a missense mutation that's just going to be a VUS moving forward. Hopefully that kind of answers your question.
I mean, I guess with the, in the T cell field or the HSC field, you have the advantage that you're looking for gain of function, and then you have a proliferative drive on top of that. So you get expansion of what you want, right? But if you from pure a basic fundamental point, if you would want to edit a gene that doesn't necessarily give you a growth advantage or a survival advantage, it's more challenging, I assume.
Absolutely. Yeah.
This topic of the guide RNA, thinking of like sometimes when you use these online tools, there are multiple of them, and for one region they suggest like 2-3 guides, and they have like different scores a little bit. Is there any way to really know which one works better? Because different tools also give different scores, or you have to just test it.
There are a bunch of different tools that'll spit out numbers that are predicted efficiency. There's nothing like testing them yourself. And so my strategy, if you want a user-friendly guide RNA design program, it'll help you make sure that you're picking like appropriate guide RNAs for your platform. I like ChopChop is what it's called. That's a really good one. But then it's going to give you like probably hundreds of guide RNAs, and how do you choose the ones that you care about? Usually you can hone in on a specific region. If you want to do a knockout, you need to go for an exon that's across all the different isoforms. And I like to go earlier rather than later in the gene. And so that's kind of my algorithm, is go to an exon that's expressed across all isoforms and try to I like to test 5 different guide RNAs with no overlap or minimal overlap. And if you test 5 different guide RNAs across a region that's, maybe 100 base pairs long, you should be able to find something that works really well. And I've never really seen any anecdotal trend of, ChopChop gave you this predicted efficiency score and that one worked better. I just think we still don't really know the rules really of what makes a good guide RNA and a bad guide RNA. And then a final thing for that algorithm too is take your candidate guide RNA sequences and just feed them into UCSF Genome browser, blast them, and just make sure that they're not showing up as homologous. All things being equal, and you probably want to pay attention to GC content, so 40 to 60% as well. That's kind of taken together my personal algorithm for designing these. Yeah. Andy, yeah.
In the scenario where you're starting to multiplex with guides, especially if their cut sites are close together, is there potential for like breaking the chromosome and causing like chromosomal abnormality?
Yeah. Yes, great question. I would not recommend Cas9 multiplexing. The double-stranded breaks, I know they've really, Cas9 is an editing platform. People really always highlight the fear of double-stranded breaks. There are some scary reports, in terms of, we always think about these off-targets being a small indel somewhere else in the genome, but most of the genome's pretty empty. And so I still have never found a guide RNA I wanted to use, but it just had like this devastating off-target and an oncogene or tumor suppressor. They're usually just in a random couple spots. To me, what's way more scary is large genomic abnormalities. And so some of the reports, my gut says the most common thing that's causing problems is a large on-target deletion. When we do those, the lamppost that we're looking under is just that like couple hundred base pairs surrounding your cut site. But you're blind to whether a large deletion happened, and there are some reports where it can happen 10% of the time. And gene clusters, if you're knocking out things that are nearby, that's kind of scary. There's also a paper from Jennifer Dowden's group that talks about chromosome arm loss following Cas9 editing with T cell manufacturing, and so rearrangements, large deletions, all of that stuff starts to be pretty scary. And there's no standard assay to find all those different flavors of off targets. And so that's why multiplexing with multiple double-stranded breaks is just a recipe for maybe disaster. We've done that just to benchmark it against our multiplex base editing, and it's toxic to the cells and causes an increase in rearrangements. But base editing, we found nothing above background that had been reported before. They're really gentle. They're really good tools. Yeah. Ed, yeah.
Yeah, I want to go back a little bit. You mentioned a little bit about tissue specificity, and it sort of struck me. Are these editors locus dependent? Like how are severely affected, or how are they affected by like secondary structure or chromatin structure, things like that. Is that an issue for tissue specificity?
There has to be some sort of impact. I don't think people have really rigorously tested these across a lot of different tissues, because then, kind of like going back to my point to Jeroen, it's like you do need like delivery. There aren't a lot of labs that are editing a ton of different, clinically relevant primary cell types. And so I don't think a really strong comparison has been done. It would, to me, be shocking if chromatin structure didn't play a role, and we're kind of in an indirect way trying to test that by doing the baby KJ strategy of taking a snapshot of a patient genome, putting it into a lentiviral vector, and then trying your correction strategies on that. We want to see is that going to recapitulate the editing that we get in patient-derived cells? So it really is just what are the DNA sequences at that region, or how accessible is that region? And it's not going to fully answer the question, but it'll at least shed light on that. I've been really amazed that you can target most places in the genome, it seems. However, that being said, most places we ever want to target are in a gene or near a gene, and so they're probably relevant to that tissue type, and so they're probably err on the side of being more open and more accessible. But it does seem like Cas9 can scan pretty much all of the genome, and there don't seem.
I guess it's in like the sickle cell experiment, right?
Yeah.
There's a lot of delivery to the liver just because of the way the biology is delivered.
Yeah.
But do you know if people have looked to see whether other tissues besides the cells are actually edited?
Yes. And so actually Tippy's folks have been doing that, and we see nothing gets your edit as much as the liver. However, I guess the caveat there is it's in the transgene cassette of a transgenic mouse. So it's and it's expressed, so it's active in all of these cell types. So it's not that perfect control. Liver is the most edited, and then there's liver and there's everything else. But heart gets some, HSC, the blood system gets some in the lung, especially if you do these early developmental time points. Yeah. But it's still kind of an unanswered question, if I had to be perfectly honest. Yeah.
When you said the nuclease comes from a bacteria, right?
Yeah.
So how well can it be tolerated by humans?
So it was found in my previous lab where I trained that we have pre-existing immunity, it was like 50% of people have pre-existing immunity to Cas9. There was this old ad I saw in like a Time magazine, and it was Listerine, and it had somebody with an open mouth, and it talked about all the different bacteria that you're killing off with Listerine. It had all of the populations, the bacterial populations that we're pulling Cas9 from. It was S. aureus, it was S. pyogenes, and it was others. So it's like we have CRISPR in our bodies already, and your cells, they're getting exposed to this to the point where there's detectable pre-existing immunity to this. The great thing is, at least for the ex vivo therapies, there hasn't been any report that this is causing a problem, because the genome editing tools, they're gone usually by the time you transplant these cells, especially in the context of like this hit and run permanent modification. However, I think the risk goes up dramatically when you're doing vivo editing. But if you do it quickly enough, the editing, people have said, will happen before your body can mount an immune response to it. So it's an important consideration, definitely. And still, especially if you're then installing, kind of to my point about what comes next, these installations, the single edit of synthetic elements, as soon as you put in non-native proteins, we're going to need to be thinking again about immunogenicity of these. So just definitely going to be a continued area of focus.
There were 3 questions on Zoom.
Okay.
Yeah.
Okay.
Sorry, Kyle. So
yeah, no, please.
Thanks from Holger. He was asking, and sorry if he missed this, but can you comment on the need for cell proliferation for the different gene editing methods?
Oh, great. Yeah, great comment. And so for Cas9 double-stranded breaks, I'll say Cas9 double-stranded breaks at base edits, they seem to occur whether a cell is proliferating or not. However, HDR seems to be the one with the biggest difference. And the conventional wisdom is the cell needs to be actively proliferating for HDR to occur. That's been challenging in HSCs, where the ones with the most stemness are typically the ones that are most quiescent. And so we do see we see effects of that, and they're really great indirect readouts to say that, oh yeah, among your HSC, true long-term repopulating potential HSCs, we're getting lower editing rates among them.
Great.
And the conventional thinking is for prime editors, and these recombinase systems, if you're actively inserting a piece of DNA with how these editors work, and so it can be done without requiring a cell population to be actively dividing. And I think that's a super important consideration for CNS disorders, and a number of others, yeah.
Great. And then another question, if the guide RNA is longer than 20 nucleotides, will that increase or decrease precision? If it decreases precision, why does that happen?
So 20 is what we've arrived at. You can shorten it. The only thing I have experience with is shortening them to like 18 and 19 base pairs, and that's going to make them less specific. The activity seemed to be preserved at that point, but I think there's also just some limit for how long you can make something and then still maintain maximal intended editing frequency. So I would bet if you made a 30 nucleotide guide RNA, you probably just eliminate all editing activity just because it's not able to interact in the right way with your genome editing tool. But I think you can go up a couple base pairs, and actually different like genome editing, like Cas9 orthologs have different guide RNA requirements, so different PAM sequences and different lengths that are ideal for each of the tools.
And the last question is from Mark Walters, who had to drop, but some of the HSC in vivo editing systems utilize short-term positive selection to enrich for edited clones. Do you think this is a safe approach, or are there concerns for, colonal hematopoiesis with proliferative expansions?
Yeah. I think anytime that you're doing something beyond just taking a patient cell, making it wild type again, there's risk. That being said, I think it's a risk worth taking given the ability for, especially the strategy that he's talking about. You could create an edit on a surface receptor that's inert and then get away from chemotherapy by giving an antibody to clear out your unedited cells. Or we're thinking about these like truncated EPO receptor expressing cells that are hyperproliferative. I think the risk might be worth it in these cases because it might allow you to dial back or fully eliminate chemotherapy, which is also going to have its own unintended risks as well. Like you're peppering the genome with double-stranded breaks, and people get secondary malignancies because of the chemotherapy regimen, not because of the safety of the edit. All of that being said, is both the strategies I just mentioned are increasing the clonality of a stem cell population. I think that has its own risks attached to it. Clonal hematopoiesis could be problematic. I also think of stem cell exhaustion. I mean, these HSC editing therapies, they need to stick around for 40 years doing what you want, not what you don't want. And so the bar for safety is extremely high. And you're also transplanting billions of cells. And so even extremely low frequency events could become amplified over time. And so, I think that the FDA is very much aware of this. I'm grateful for the CAR T cell space that's kind of the tip of the spear for like heavily engineered cell-based therapies. At the end of the day, I think if they work well and they have other advantages, then there will be adoption of them, but it's just going to take some time.
Thank you.
Okay.